Okay, I'm going to go ahead and get started. So, hello everyone. Welcome to the second to last installment of the SynCell 2021 speaker series. My name is Jacqueline Delora. I'm a postdoc at the Max Planck Institute for Medical Research in the Cellular Biophysics Department in the lab of Professor Spath. A couple of announcements before we get going. There is one more seminar series next Monday, on the 17th of May, that will launch us into our conference. And as you can see here, the registration for the virtual conference is already open. You can go to syncell2021.unm.edu to register. And also our poster previews will begin tomorrow, and you can find more information at the website as well. So today we have two excellent speakers here, Professor NEAL DEVARAJ and Dr. Emiliano Altamira. It is my pleasure to introduce our featured speaker, Professor NEAL DEVARAJ, who will start the seminar today. He did his undergrad at MIT and doctoral studies at Stanford University, where he earned his PhD in chemistry in the labs of Professor James Coleman and Christopher Chidsey. He then was a postdoctoral fellow at Harvard Medical School in the lab of Professor Ralph Weissleiter, and subsequently joined the faculty at UCSD. Professor DEVARAJ focuses on the design of chemoselective reactions to address problems in bottom-up synthetic biology and molecular imaging. I will turn it over to Professor DEVARAJ now, and remind the audience to submit your questions through the Q&A button on your Zoom interface. So thank you, Dr. DEVARAJ, whenever you're ready.
Yeah, so can you see the screen?
Yes, it looks great.
Great. Well, yeah, I just want to first thank the organizers for the invitation. And yeah, of course, it's a shame. Was looking forward to the large conference that we're going to do, but of course, this is a necessity. And really great that we have this opportunity to get together and talk about, I think, a topic that's of interest to all of us here at JAR, synthetic cells. So I'll share with you a little bit today what my lab's working on in trying to mimic lipid membranes. So, I think this is not something I need to convince the members of this audience, but I think one of the really major questions in science today is, although we have a lot of knowledge and a good understanding of how the molecules of life work, trying to understand how these molecules come together and organize themselves to create structures, namely cells that have the fascinating properties of life. This is still really quite unknown. And so there's a lot of work in trying to understand how do we sort of transform, if you will, chemistry to biology. And this is something that my lab is really quite engaged in right now. And we really think that one of the main issues is compartmentalization. So as you know, compartmentalization is an absolute requirement for life. Compartments couple metabolism and replication. They also localize entropy-reducing processes, and the major way in which cells, which are the minimal unit of life, compartmentalize themselves either from the environment or internally in the case of eukaryotic cells is through the use of lipid membranes. And so lipid membranes are really kind of a remarkable material. It's really fascinating to think that a 4 to 5 nanometer thick bilayer of amphiphilic molecules that are held together by the hydrophobic effect and self-assemble is what's really defining the boundaries of life. And lipid membranes serve a number of important roles in cells in promoting and protecting chemical reactions, controlling the exchange of matter and energy with the environment, defining boundaries, and maintaining ion and pH gradients. And I think if we're, really thinking then about mimicking life, it really is the one place to really think about is trying to mimic the compartment of life. In that case, that we have to sort of gain a better control then over lipid membranes. So, the typical way in which many people reconstitute lipid membranes and form vesicles is by hydrating a thin film of dried phospholipids, and that will result in membrane-bound vesicles. But these are really quite simply passive containers in many ways. I mean, we can be forgiven for thinking about them as sort of static compartments. And instead, the vesicle itself, the membrane isn't that interesting. It's really the stuff that's inside that's really interesting. But if we compare this to membranes in living cells, well, really nothing could be further from the truth, because membranes in living cells are highly dynamic, far from equilibrium structures that are undergoing constant formation, degradation, growth, fission, etc. And so really one of our goals in our lab is to try to be able to emulate lipid membranes so that they're more dynamic and lifelike. Now, the idea of mimicking lipid membranes isn't completely new. About 40 years ago, the late Jano Svendler came up with, I think, a wonderful term, membrane mimetic chemistry. And his idea was to create systems that mimic aspects of biomembranes. But at the time, 40 years ago or so, the really emphasis was on trying to use amphiphiles and self-assemblies for practical applications. Could you control the rate of organic reactions within these micelles, for example? Could you enhance solar energy conversion, do targeted drug delivery? And in fact, at the time, he really sort of went out of his way to make sure that it really wasn't an area for faithfully reproducing the features of biological membranes. But I think now, 40 years later, we can revisit this concept of membrane mimetic chemistry and actually expressly go after trying to mimic the biological properties of membranes, and think about using the cell as inspiration to develop minimal chemistry so that we can mimic properties like growth, division, transport processes, and even mesophase transitions. And simultaneously, as we start developing these and mimicking these membranes, we're going to be developing a suite of tools and gain a much better understanding of how to manipulate lipids. And I think these can be fed back into living cells to help us understand the roles of lipids in life. For example, what are the specific functions of particular lipid species? We make thousands of different lipid species. What are the functions of each of these? Or can we potentially develop tools to image and track lipid species in cells? So, as I mentioned, when we first sort of started our group, the typical way of generating lipid membranes was to hydrate, thin lipid membrane films, or there's more sophisticated techniques that use phase separation. But in terms of cells, well, cells build their lipid membranes de novo, and they do this through coupling chemistry with self-assembly. And it's really kind of typified by the so-called Kennedy de novo synthesis pathway, where you build up the acyl chains by fatty acid synthesis, and these are coupled to membrane bound to polar head groups by membrane bound acyl transferases. And so we were interested in trying to mimic the growth of lipid membranes, but we felt that this was really rather complicated to try to reconstitute the entire pathway. And so we began to look for other ways in which we could try to mimic lipid membrane synthesis. And really quick refresher for why are we interested in phospholipids in particular in trying to mimic lipid membranes? Why are phospholipids a privileged molecule? Well, if we look at a phospholipid at its head group in two acyl chains, the cross-section of the head is about the same as those of the chains. And so it has a cylindrical-like shape to it, and it'll pack to form more planar bilayers. In contrast, if you were to lop off one of these acyl chains, you would form a more conical shaped molecule, and this is going to want to pack to form micelles instead. And so knowing that, we were drawn to another lipid synthesis pathway in biology known as the land cycle or remodeling pathway. And this pathway already formed phospholipids are cleaved by a phospholipase, forming a lysophospholipid. It's coupled to a thioester derivative of a fatty acid by a lysophospholipid acyl transferase. And so what was inspiring to us about this is that you have now a process by which you have two single chain precursors, neither of which have that, the right shape to form membranes, coupled together in a single coupling step by an enzyme to form a membrane forming phospholipid product. And so we thought, this kind of idea of just putting two chains together, that's something we could mimic using chemistry. And so, really the concept is, can we couple together these two single chains and form a phospholipid, and that presumably would lead to a bilayer. What's kind of interesting is this sounds simple in principle, but it would involve so-called de novo membrane formation, that is membrane formation in the absence of pre-existing membranes. So I think many of us here are familiar with cell theory and the statement that all cells come from cells, that I guess the people in our community are trying to, prove this is not true. And we know it's not true, from an evolutionary perspective. But in 1980, Gunter Blobel, in a very famous PNAS paper, extended this concept to membranes, stating that all membranes come from membranes. And that's a pretty bold statement, but to my knowledge, there's not really one verified example of biology making a membrane, in the absence of a membrane. All these membranes are coming from membranes. And this kind of makes sense because membrane lipid synthesis is catalyzed by a series of membrane-bound enzymes. And these membrane-bound enzymes require pre-existing membranes for proper folding and function. So there's kind of a chicken and the egg paradox here. And so it does kind of lend itself to this question of, well, can de novo membrane formation occur, period? And so the very first example we did is we decided to utilize a very selective chemical reaction, sort of commonly known as click chemistry. And we chose this reaction because it's highly yielding and also works very well in water. And you can now couple together this azide and alkyne and form this artificial triazyl phospholipid. And in water, you can actually watch membrane formation take place. So this is a dye that's staining membranes. And when you add the catalyst, you get nice fields of vesicles. And after 12 hours, you're left with nothing but vesicles. You've consumed all the reactant. And this can be done with other reactions. This is an example called the native chemical ligation, where what's nice about this is you actually use the thioester precursors, similar to how biology makes lipids. And here, both these precursors, in fact, form micelles. And on the right here, I'll show you this video where we mix these together, and you can see the formation of these sort of tubular structures that grow with time, and then they grow sort of longer at peers. And then after the reaction nears completion, they become more spherical in nature. And just one last thing here, we very recently even shown you can make natural phospholipids using similar kinds of concepts. A really great postdoc in my lab, Luping Liu, showed that if you actually make thioesters with thiocholine and oleic acid, those positively charged now thioesters can react and acylate natural lysophosphatidylcholines, forming natural phospholipids. And these can also spontaneously form membranes de novo. And they can also support ion gradients. So, most of my lab has been, for the most of the time, we've been really focused on sort of mimicking phospholipid vesicle formation. And these phospholipid vesicles are great because they're compatible with biochemistry and they can maintain ion gradients. But when we start thinking about, utilizing these systems as sort of synthetic organelles or bioreactors, we're faced with some issues. So, yes, the phospholipids maintain ion gradients, but that's because they're impermeable. And that impermeability we need to have to tackle if you want to start getting, reactants into the vesicles and waste products out. And so that means you really need a sophisticated transport machinery system. And obviously biology gets around this because they've evolved, biology's evolved over billions of years, really sophisticated transport proteins. But these are often very challenging to reconstitute in vesicles. Now, recently in our lab, a few years ago, in fact, we started exploring an interesting class of amphiphiles. We actually pretty much have stumbled across. And these are these non-ionic amphiphiles, glycolipids that have galactose head groups. And it turns out that these single chain amphiphiles, surprisingly, will form giant vesicles. And because they're single chain amphiphiles, the permeability of small molecules to these vesicles is greater than phospholipids. In fact, you can do biochemical reactions like rolling circle amplification reactions inside these vesicles and feed the precursors from the outside. And while we were exploring these, amphiphiles, which can be made from thioester coupling or even from amide coupling, we started thinking about ways in which we could try to reconstitute membrane proteins. And so a student in my lab, Ahanjit Bhattacharya, began exposing these galactolipids to a detergent called iGPAL that's often used for membrane protein reconstitution. And in doing so, he noticed something really quite surprising. So when he mixed these two together, rather than seeing vesicles, he noticed these droplets in solution. And it was very spherical structures. If you put, dye that goes in the droplets, they'll phase separate with time, and that can be easily, observed that you can see that. So it definitely is a phase separation. And the droplets can be made through a variety of ratios of iGPAL and the galactolipid. So if you have a lot of the galactolipid, you get vesicles. If you have a lot of this detergent, you get micelles. But a lot of ratios, you'll get droplets. And so when we first saw this, we thought, well, we have these two non-ionic amphiphilic molecules. They're very greasy. Probably we're making oil droplets. And you notice that there's a galactose head group here. So we took a fluorescent protein that binds to galactose and added it to the mixture, thinking, okay, well, maybe we can decorate, the periphery of these droplets. But when we did that, instead of seeing the periphery light up, we saw the entire droplet be stained and labeled, the interior included. And so this forced us to rethink, what we potentially had on our hands. And so Anjit, to his credit, actually collaborated with the Sunil Sinha lab in physics here at UCSD and started teaching himself a variety of characterization techniques. They're often used in soft matter physics. So he, did small angle X-ray scattering, neutron scattering, cryo-TEM, cryo-SEM, and eventually came to the conclusion that we don't really have droplets. But in fact, what we believe is happening is there's actually a phase transition occurring, and you're going from the lamellar phase, which is sheet-like and what we typically think of when we think of lipid membranes, to what's known as a bicontinuous sponge phase. And so this bicontinuous sponge phase is, rather than sheet-like, is a sort of three-dimensional network of bilayers and water channels. And so it's bicontinuous, it's throughout the whole droplet. And that also explains why these proteins could stain the interior of the droplet. Is there's basically water channels and pores all around the interface between the two phases. And so, as long as the molecules are small enough to go through the pores, which are about 4 nanometers in diameter, and we know that from the X-ray scattering data, the molecules can enter into the droplets. And so these droplets can be made by different chain lengths of galactolipids and different kinds of surfactants that are similar to iGPAL, like Triton X, for example. And one thing I should point out that I think is very interesting is that these kind of non-lamellar bicontinuous phases have actually been observed in cells in many different organelles. For example, in plants, there's an organelle known as a prolamellar body that's quite interesting. But also, people have shown that you can overexpress certain membrane proteins and trigger the formation of non-lamellar phases in the endoplasmic reticulum. And also, quite, I think, topical for us right now, actually, these kind of phases that are bicontinuous are often seen in cells that are infected with viruses. So this is actually a Vero cell that's been infected by SARS-CoV, and the ER has now undertaken this sort of what they would call a cubic phase morphology. And in fact, what you're seeing here in these dark objects are virus particles. So it's thought that the cubic phase or the non-lamellar phase is actually serving as a depot sequestering viral particles. So in thinking about this, we started realizing, well, maybe we could potentially utilize these droplets as synthetic organelles. So we had to ask a few questions before doing that. One, how stable are these systems? So it turns out that, in fact, they're very stable to different pH. So you can scan pH 2 to 10, different salt concentrations, stable to magnesium and calcium, for example, stable in seawater, stable in cell media, they're even stable in cell-free transcription translation systems like the PURE system. And a wide variety of small molecules, fluorescent dyes, for example, can readily get enriched in these droplets. The story here is very simple. Pretty much the more hydrophobic the dye is, the more it gets enriched in the droplet phase. I think more interesting is protein sequestration. So it turns out that galactophilic proteins like LEC-A, LAC-I, or galectin-3 will all get enriched in the droplets. And so in this case, we have fluorescent versions of these proteins, and we can see, even 100-fold enrichment compared to the bulk of these proteins in the droplets. And we can actually even reach millimolar concentrations of droplets. We estimate millimolar concentration of proteins we estimate in the droplets. So one way of seeing this is we can actually add the droplets to the cell-free transcription translation system, like the PURE system, and also add the DNA encoding for a green fluorescent protein fused to LEC-A, which would bind the galactose head groups. And these, the droplets in the PURE system are mutually compatible. So we can actually watch then protein expression in the presence of these droplets. And so this is phase contrast. You can see the droplets, hopefully. And then now we add the DNA and watch, and you can see the droplets just light up. And that's because as you're forming this GFP-like A, it's basically immediately getting sucked into these sponge droplets. And so it's a kind of a really nice way of sucking in proteins that have affinity. But of course, the idea of trying to like fuse a protein of interest to a lectin is not really that convenient. And so we were looking for other ways in which we can program what goes into the droplet. So GFP on its own doesn't partition spontaneously into the droplets. And so we thought the fact that the even though it's a non-lamellar phase, it's still consisting of these network of bilayers. Perhaps one way we could program what goes in is by doping in functional amphiphiles at a low percentage. So as an example, we can dope in a low percentage of a nickel NTA binding phospholipid, where now a polyhistag protein would bind in the presence of nickel to this phospholipid. When we do that, now histag GFP goes into the droplets. If we don't dope in that lipid, we don't get any partitioning. And this game can be played over and over again. So you can take a mannose binding, mannose containing lipid, dope that in. Now mannose binding lectins go in. You can put a positively charged lipid in. Now negatively charged oligonucleotides go in. And you can take a biotinylated lipid, and now fluorescent streptavidin goes in. So this turns out to be a really convenient way to dope in various compounds and program what proteins go in. And we're exploring this even more now in our lab with other types of affinity schemes. So then the question is, well, can we actually then use these droplets to act as artificial organelles and mimic functions that occur in organelles like lipid synthesis, protein degradation, or sequestering or protecting biomolecules? So before doing that, a postdoc in my lab, Henrique Niederholtmeier, was interested in asking, well, if we put something into the droplets, are they static or are they mobile? Are things moving around, the lipids, the proteins? If we want to have reactions to occur in the droplets, we would kind of like things to be mobile. Now, we saw from just watching these droplets that they seem to be liquid-like, and you can see this from the droplets just coalescing with one another. But Henrique really wanted to get more quantitative about this, and so she did put in various types of fluorescent lipids or fluorescent proteins into the droplets, and then did some fluorescence recovery after photobleaching experiments. And so you bleach a section of the droplet and then watch to see if it recovers. If it doesn't recover, that means things are really static. And indeed, in both cases with lipids or proteins, it does recover. And what it basically tells us is that these entrapped materials are indeed mobile and can interact with one another. And so then, one other question we had then, in terms of particularly for mimicking lipid synthesis, is can we host membrane proteins in these droplets? And that turns out to be really straightforward. You can dissolve your membrane protein in the amphiphile detergent and then just simply add the galactolipid to those micelles, and that'll form the droplets. And so we did this with a very well-known membrane protein, cytochrome C oxidase, and you can see this is fluorescently labeled cytochrome C oxidase. It goes readily into the droplets, and then it's still functional. It can still oxidize ferrocytochrome C. And I think what's kind of neat is that, yes, we can monitor that by absorbance, but because we have such a high surface area of bilayers, we can actually incorporate quite a bit of cytochrome C oxidase in these droplets. And so you can actually just by eye see the difference as you oxidize ferrocytochrome C. We also looked at more lipid synthesis type reactions. So here we've reconstituted diacylglycerol kinase, and this is a membrane protein that phosphorylates diacylglycerol. And now we can take a lipid substrate, and in the presence of magnesium and ATP, we can see that we can phosphorylate that lipid substrate. And so this is something that we're using as a foundation to expand on the idea of trying to can we actually reconstitute more complex lipid synthesis pathways into these droplets. We can localize peptides and proteases for degradation and see how that impacts the kinetics of degradation. So here we've taken droplets that can bind histag proteins. We take a peptide substrate that has a coumarin that when cleaved fluoresces. And it turns out that peptide substrate on its own will localize to the droplet, maybe because of this coumarin. And then we can also take a protease, cathepsin K, that's histagged, and this will also bind to the droplets. And so now if we just have these two in the bulk phase at these low concentrations, we see very little hydrolysis of the peptide. If we add the detergents or add droplets that can't bind cathepsin K, we see a moderate increase in the degradation. And we think that's because we're doing a better job solubilizing this peptide. But if now we add the droplets that combine both the cathepsin K and the peptide, we get this really nice increase in the fraction of the peptide hydrolyzed. So these droplets could be a really convenient platform for studying how co-localization, compartmentalization can affect the rate of biochemical reactions. So finally, we were interested in thinking about ways in which we can gain more control over, what and when the proteins go into these droplets. And so to do that, we were interested in using light then as a way of having a stimuli control over protein partitioning. And so we took advantage of these two proteins, photoreceptor phytochrome B and phytochrome interacting factor Phi B and PIF. And these are plant-based proteins that when you shine 660 nanometer light on them, they interact with each other. But when you shine 740 nanometer light, they separate. And so Henrique and Hanjit took these droplets and they localized Phi B into the droplets and then created a PIF GFP. And then now if you shine 740 nanometer light, these two would be separated. But if you shine 660 nanometer light, it should drive the PIF GFP into the droplets. So that's a video I'm going to play here where you can kind of make out the outline of the droplets here. This is the wavelength of light. So initially we're starting with these separated, and then we'll turn on the 660 nanometer light and the fluorescent proteins go in, and then you turn on the other light, they go out, and then this can be repeated over and over again. It's a reversible interaction. So with this in hand, we were curious whether or not we could try to use light to sort of drive a protein into the droplets and protect it from sort of a protease that would be outside. So it's a little bit kind of different now from the previous experiment where we're co-localizing the protease with the target. Now we're trying to separate them. And so we use as a protease ClipXP, which is this really huge complex, about 600 kilodalton, about 10 nanometers in diameter. If you recall, the pore size of these droplets is 4 nanometers. So this protease can't get in. In fact, if you fluorescently label ClipP, you can see it just stains the periphery of the droplet. It can't get inside. And then we can tag the PIF GFP such that it'll be recognized by ClipXP. And so if we just have 740 nanometer light, so these two are separated, we see this nice rapid degradation of the target. But now if we intersperse in 660 nanometer light, you can actually see a really decrease in the rate of degradation. And this line here, so what we've done here is we have it in the droplet, slowly degrading. Then you shine 740 nanometer, expel it from the droplet, it gets much faster. Then you go back in the droplet, out of the droplet, back in the droplet, out of the droplet. So again, this is a nice little handle, I think, to have, and it really shows, I think, the degree of control one can have over performing experiments studying how co-localization affects biological reactions. And I just want to end by saying that one thing we're really excited about is we were recently supported by a MURI grant to look more carefully at these droplets. And so we've got this really great team of investigators that are coming together to ask various questions about how these droplets can be used to organize biology. And with that, I'll wrap up and thank the organizers again for the invitation. And I really want to thank my group. It really shares a passion like myself for science at the discipline, at the interface of chemistry and biology, the interface of disciplines. And I want to thank all our sources of funding, and I'd be happy to answer any questions.
Great. Thank you so much, Professor Devaraj, for that amazing talk. We do have a few questions already from the audience. I myself have two just quick technical questions to ask you. The first, are the droplets temperature sensitive? Did you look at that as a handle?
Yeah, that's a great question. I mean, I think if you go too low in temperature, they'll start to, aggregate or have problems. But we didn't really look at that very carefully. But it's a very good question.
Okay, and then I guess kind of leading into that, I'm just wondering if you've thought about multi-compartment architectures where you might be able to recapitulate, say, like the periphery versus membrane trafficking that we actually see in real life.
Yeah, that's really a great question. We've definitely been thinking about it. I mean, one kind of wild idea I've been thinking about is, since we can now reconstitute membrane proteins, could you actually, having like lipid synthesizing reactions inside these sponge phase droplets to make lipid droplets. So if you look at sort of the models in which lipid droplets are thought to be made from the ER, you have a sort of ER network of bilayers, and then you're making these triacylglycerols, and these triacylglycerols are supposed to sort of bud off and form these droplets that are coated by a monolayer of phospholipid. And so that's actually something I'm kind of really interested in, is can we reconstitute those proteins into these droplet systems and see if we can actually make lipid droplets that way.
Yeah, great, thank you. Okay, we have a question from Jan Steinkühler. He asked, did you look closer at the dynamics of sponge droplet fusion? I could imagine that it would require rearrangement, fusion, or fission of the sponge membranes and be more complex, slower than fusion of normal droplets in a continuous phase.
That's a great question. I mean, we didn't look at it very carefully, but it's something that we've been really interested in, is can we, what can we do to control the fusion, or even can we have these droplets undergo fission as well? But even just fusion rates, that's something that's really interesting. And one thing we've been looking at a little bit is how does the loading of proteins into the droplets, or potentially decorating the droplets with other types of biomolecules like DNA, how does that affect the coalescence and fusion rates? But the short answer, we didn't do a very good job quantitating that, but it's something that we want to do more of looking forward.
Great. Next question starts with a statement. Galactolipids can self-assemble into rod-like micelles in water. By partly removing the agapulp, can you morphologically reconfigure or elaborate the droplets with the cytoskeletal-like galactolipid filamentous network? And this is from Professor Steve Mann from Bristol.
Yeah, that's a great question, Steve. Thanks. So one thing I should point out, and this is really quite fascinating, is glycolipids can form stiff rod-like structures. But what's interesting is you mostly see that from glucose the exact same lipids that we worked with. Instead of galactose being a head group, if glucose is a head group, you get these really rod, stiff-like structures, and that had been known for many years. And so it's really, quite fascinating to think about just changing that one OH has now made it so that when you have these galactolipids, you can make vesicles. But it is true that we have also observed, and we've published on this, that even the galactolipids can undergo transformations into rod-like structures. So I think we played around a little bit trying to, either, mix in these other types of lipids that can form rod-like structures. But I think typically when we have these mixtures, we were never able to, I guess, see, if I'm understanding what you're getting at, the idea that you'd have sort of a vesicle or a droplet and then have more cytoskeletal-like elements coexisting at the same time. I guess that is kind of a tricky thing in terms of having, multiple amphiphiles coexisting into separate phases at the same time. I guess, biology is able to do this. I think that's what's kind of, remarkable. You have so many different membranes coexisting and sometimes even different phases coexisting. But that's something we haven't been able to gain control of over yet. But thanks.
Good. Matt Good says, very cool work, Neil. Have you tested strategies to alter membrane surface area to volume ratio to manipulate droplet morphology? For example, through fusion with small vesicles, alteration of osmolarity, or feeding amphiphiles into the solution?
Yeah, a great question. And I think our main approach, all those techniques would be interesting to try. And I think the osmolarity approach in particular is very interesting. As chemists, we've been really focused on, synthesizing lipids. And so where we've been really kind of emphasizing our efforts is in trying to expand the membrane through lipid synthesis to try to play with that balance between the surface area and volume to try to get shape transformations. So where we're at now is, we have previously observed, and I think, we've sort of seen sometimes when we feed the precursors and generate additional membrane, that we'll see sometimes shape transformations, but it's not very well controlled. And so I think one of the issues is that in the current systems that we're working with, we're always relying on single chain amphiphiles as the precursor. And the single chain amphiphiles, as you know, they'll be detergents. And so you kind of have this problem where if you add too much, it starts disrupting the membrane, you start creating pores, So one of the things that we've been working on more now is can we develop ways in which we feed not single chain amphiphile, but like non-amphiphilic precursors that eventually get converted chemically to phospholipids or phospholipid, non-canonical phospholipid. And that's how biology synthesizes membranes. And in fact, we have a paper that's about to come out soon, but it's on bioRxiv, where we've shown that we can actually reconstitute a type 1 fatty acid synthase that can take acetyl-CoA and malonyl-CoA and make it into palmitoyl-CoA, and then that gets immediately converted to phospholipid by one of our native chemical ligation type reactions. And so that's kind of nice because in that system, you can actually feed acetyl-CoA and malonyl-CoA in to try to generate additional membrane area.
A sort of loosely associated question from an anonymous attendee. They ask, is it possible to tune the pore size by addition of large lipid head groups or long chains of PEG?
Yeah, that's another fascinating question. So we initially tried to play around, and I kind of briefly showed up different chain lengths of glycolipids. And with the X-ray data, we didn't really see too much of a change in pore size. But just recently, and I mean, just last week, we had a meeting with some of our collaborators, and it turns out that one way in which we might be able to change the pore size is kind of what I think what the member is asking, is by changing the amount of phospholipid we dope into the droplet. So it does appear that by changing the ratio of the phospholipid we dope in, this is actually can have a pretty severe effect on the pore size. And so, at least from the X-ray data. So I think that'll be really exciting. That's something we've been kind of on the lookout for, is we'd love to be able to control the pore size and do it in a way that which we could actually have stimuli control. It would be really interesting.
Great. All right, last question from Himanshi Singh. They say, hi, great talk. Have you considered the application of sponge droplets into food, sensor, or packaging, such as absorbing the toxins if detected in the packaging, possibly a bit of an industrial use?
That's a really nice idea. No, we haven't really thought too much on, I would say, the industrial side. We have, thought a little bit about drug delivery or, sort of, sequestering proteins, things like that. But, one of the challenges there is, of course, the cytocompatibility of the droplets, which, is a little tricky with all the detergents. So we're actually exploring other systems that might be more biocompatible, like other detergent systems. But I think, to your point, great idea, because I think if you're thinking about things from the more industrial or packaging standpoint, now it doesn't necessarily have to be so compatible with cells. So yeah, I mean, the application side is something we're really interested in. And again, I think by exploring other kinds of detergents that can form these systems, and there are others that are out there. In fact, for example, Fred Menger and some other people have looked at systems that can form these kinds of sponge droplets. But that they sort of left it at that. They sort of said, okay, look, we saw it makes a sponge droplet, and then they sort of moved on. And I think now, I think what's cool is we have this opportunity as a community with synthetic biology, where it is, we can start using these droplets as potentially organelles, programming proteins that go in, making these sort of more interesting systems. And so we're starting to revisit, those kinds of systems. And some of these, I think, will be more potentially, maybe appropriate for certain applications in the presence of cells, for example, or in packaging or things like that.
Yeah. Indeed. Okay, one last question actually now. So Gabriel Lopez says, beautiful work. Is there a means of controlling the size of the lipid sponges? Your last slides implied an interfacial boundary layer.
Yeah, I mean, that's a great question. And I think one approach might be to like controlling that interface. So one idea would be to prevent the coalescence by decorating the surface with a repulsive molecule. And so one thing that we've been exploring, actually, Henrike Nierholmeyer, who did a lot of this work that I talked about, she's now at the Max Planck as a group leader there. And so we've been working on actually developing a manuscript where we're talking about modifying the droplets with DNA. And so she's actually gotten some really nice data on that. And so one thing we've seen, we can actually, if you make the DNA large enough that it doesn't actually go through the pores, you can decorate just the surface of the droplets, and you can see that. But if you have fluorescent DNA, just the periphery lights up. And so this could offer a way in which you can now start potentially controlling the coalescence, maybe controlling the size, potentially even programming like what gets decorated on the droplets. So we're really excited about that direction as well.
Great. Well, thank you for the excellent Q&A session and your talk today. We really appreciate it.
Yeah.
Okay, so